Okay, hello everyone. I hope you're all having a good day. Um, my project is based in Yi Chen's lab at UCSD, along with Ming-Xuan Kai as my grad student mentor. And this project lies at the intersection between DNA nanotechnology and drug delivery. So one thing I want to say before I get started is if I want you to look at a particular part of the poster, I'll show you with my mouse. And if I'm not pointing to anything in particular, you can just look at me and listen to what I'm saying. So the objective of this project was to create a DNA nanostructure modeled after herpes SimplexVirus1 and use it as a drug delivery tool. So there's kind of a lot to unpack there. So let's just start with what we're modeling our structure after. So the herpes virus can be, for simplicity, sectioned off into 3 different parts. So we have the envelope, the tegument, and the capsid. And since our goal is to use this for delivering materials, we are modeling our structure after the part of the virus that houses its payload that it will deliver into cells that it infects. And that's the capsid. So the capsid is in the shape of an icosahedron. It measures between like 120, 125 nanometers across. And an icosahedron is a roughly spherical shape that's made up of 20 different triangles. So our first goal here is to create those triangles. And using DNA, there's a lot of different ways we could do that. But the technique that we decided to employ for this project is called DNA origami. The idea behind that is that you have one super long strand of DNA called a scaffold strand and a bunch of tiny little strands called staple strands that kind of form the shape and the structure of the scaffold strand, make it look like what you want it to look like. So what that ends up looking like in our project is kind of like what this model looks like over here. So the scaffold strand is represented by these blue blocks over here. The actual strand would be running along the perimeter of these blocks, and the staple strands are represented by these green and purple arrows in here, and they're just making sure that the triangle holds the structure that we want it to hold. These orange lines up here they don't represent sections of DNA. Also this blue line down here, those are just ways to mark those sides in figure C down here. So these 2 orange sides correspond to that side, and that blue side corresponds to that side. What we would expect this to end up looking like is what you can see down here in the bottom left corner. These are triangles. You can see there's equilateral triangles with what looks like little cuts in the middle. That's what we're looking for when we're creating our structure. So now we have triangles to make up this icosahedron, but next we have to find a way to connect them. One of the difficulties there is that we can't guarantee that all of these sides have the same DNA code. And the way the issue with that is that each of these sides needs a complementary edge from another triangle to connect to it. And if they're all different, then we would need a different edge for each triangle. And that minimizes the amount of ways that the triangles connect to one another, and that reduces our yield. So the way that we're dealing with that is we have a universal edge at the end of each triangle. And one side of that edge connects to the triangle itself. It's made up of staple strands, as you can see here in red. And the other side is the same on every triangle. So what that does is it allows us to connect any side from any one triangle to any side from any other triangle. And that maximizes our yield. So now we have a triangle and we have a way to connect them. But now we need to find a way to encourage those triangles to form the structure that we want it to form. Because there's a lot of different ways these triangles can connect to one another. They could just, form a plane of triangles like you see down here, just a bunch laying flat. So the way that we're doing that is we're adding a hinge between the triangles. And what that hinge does is it controls the angle between 2 different triangles and it makes sure that it's the same angle that triangles connect at in an icosahedron. So that angle is about 138 degrees. So what this hinge does is it makes sure our angle of connection is about 138 degrees. Now, so we have a way to create our triangles, a way to connect them, and a way to encourage them to form the three-dimensional structure that we want to form. Now, this is really important because this is a brand new way to create three-dimensional structures like this. DNA origami has been used in the past to create wireframe structures of complex polyhedra. So think of like, a construction site. There's the scaffolding. You can kind of see what the shape of the building is, but there's not the actual building there. That's what it's been used for in the past. It also has been used to create two-dimensional arrays of faces like this down here, a bunch of, shapes. It's been used, this is my favorite, to make like drawings. Like you can draw a happy face with DNA origami, and it's super sick. But there's no reliable way to create a three-dimensional structure with a filled face. And this has potential to be that. And this is important for drug delivery because it solves the three big issues with drug delivery, being biocompatibility, cell targeting, or it has potential to solve the issue of cell targeting, and control over the structure. So biocompatibility, this is made out of DNA, which is a biocompatible material. No issues there. Control over the structure. This scales to pretty much any shape that you can think of, as long as you can, as long as it's a polyhedron. As long as you can make it with a flat shape, you can make it with this. And what was the other one? Cell targeting. So this is I think probably this is my favorite and probably the most important part of this, is you can attach materials to the face of each triangle, and some of the things that you can attach are proteins that might help the structure deliver the drug to particular cells. And that's super important for effective dosing and for reducing nasty side effects of drugs. So this is huge. And our future work is to take this design and make any necessary adjustments to it, get into the lab and actually build it, see how well it forms, test its drug delivery efficacy using various drugs, and test its cell targeting ability by adding surface proteins from HSV1. Okay, I have a little bit of time, so I'm sure one question on your mind is why we actually chose HSV1. The reason we chose it is because it's super good at targeting cells and bypassing the immune system. So it has a bunch of different ways of doing that, and we're hoping that by using surface proteins from HSV1, we can mimic those cell targeting abilities. It can also target cells that are super difficult to get to, like neurons. And we're hoping that using that, like any drug involved in a neurodegenerative disorder or anything that involves neurons, we're hoping that this could help with that or something related to that. I'd like to lastly just acknowledge the GEAR program in UC San Diego, Jacobs School of Engineering, for allowing me to do this, as well as Yi Chen for graciously taking me into his lab, and Ming Xuan Kai for helping me out very much with this project. Thank you.